Dear friends, here we are going to study about vulva formation in C. elegans. It is an organism and it comes in the topic development biology. Vulval formation in C. elegans, which is also called CAENORHABDITIS elegans. It is a multicellular nematode. The other name for the nematode is Aschelminthes, and commonly they are called roundworms. They look like a round body shape. And the C. elegans can be of two types, hermaphrodite C. elegans and male C. elegans. Hermaphrodite C. elegans will be having male and female sex organ in a simple body. Hermaphrodites can be viewed mostly simply as a female that produces limited number of sperms. Female can reproduce either by self-fertilization by using her own sperms or by cross-fertilization after the transfer of male sperm by mating. This is a C. elegans simple diagram in which these two dots are showing male organ and a female sex organ. One black dot is surrounded by an orange color. This is a female organ in one body, in one C. elegans body. C. elegans, as I told you before, it is a nematode and Aschelminthes, and in this type of fertilization can be seen: self-fertilization, cross-fertilization. Cross-fertilization can be like this, sperm moving from one body to other. Can C. elegans infect humans? Pseudomonas aeruginosa is an opportunistic bacterial pathogen of humans and the most commonly studied pathogen in C. elegans, where it causes a lethal infection of intestinal epithelial cells of the body. What is C. elegans used for? As it is a nematode, its use in large numbers can be easily screened for effects of novel drugs on complex processes involved in human disease. It is useful for study of aging processes because organism passes through several distinct phases of life which can be discovered physiologically or genetically. This is the part 1 of the vulval formation of development biology. C. elegans is used in genetic engineering. Why it is used in genetic engineering? RNAi, which is called RNA interference, is biological process in which RNA molecules are involved in sequence specific suppression of gene expression by double-stranded RNA through translation or transcriptional repression, as it involves sequence specific suppressor of gene expression by double-stranded RNA. RNAi, which is RNA interference, can be used for C. elegans genome manipulation. Transformation of C. elegans. E. coli has a gene of interest. C. elegans is allowed to feed on them, and in this way the C. elegans get transformed by gene of interest. Look at the word vulva. Here it is written 22 cells, female sex organ. Vulva is a female sex organ present mid-ventrally in the C. elegans on the ventral side, just mid-ventrally. 22 cells are there in that. The vulva is a part of female sex organ that consists of 22 cells. Vulva serve as structure through which sperm enter and fertilized eggs leave the gonads. Gonads, you know, male and female gonads, as we see in different organisms, and here vulva is a female gonad. Vulva development in C. elegans involves a network of intercellular signaling. Intercellular signaling is between the different cells. Here, for example, the 4 cells are shown. C is cell, and number 1, 2, 3, 4 tells 4 cells. When the signaling takes place between the cells, it is called intercellular signaling. If it takes between cell, it is called intracellular. So here it is intercellular signaling. And the second process, signal transduction take place in there. Transcription regulation also take place in there. Vulva is necessary for egg laying. The part we are talking about, the female part. Vulva is present mid-ventrally in the C. elegans body. Vulva is a part of female sex apparatus. Vulva has 22 cells, as I told you before also. This is the box which is showing the ventral and the dorsal side. The lower part, which is toward the land when the animal is lying, it's called ventral side. Back part away from the land is called the dorsal part. Here in the diagram you can see this is the ventral part and the upper part is the dorsal part. Life cycle of C. elegans. Life cycle of the C. elegans depend upon the temperature. So it is called life cycle is temperature dependent. Here 3 temperatures are shown and the C. elegans develop accordingly. At 16 degrees centigrade, C. elegans takes 2 to 3 days to develop as adult. At 20 degree temperature, C. elegans take 3 to 4 days to develop into adult. At 25 degrees, C. elegans take 5 to 6 days to develop into adult. This is the part which I want to show you. This full part I will be showing you in this organism C. elegans, which is diagram is drawn over here. What are the different parts? This is gonad, the green color bar-like structure is representing gonads. Under that or beneath that is anchor cell, which produces a ligand which activates the VP cells. VPC cells means vulva precursor cells, which are 6 in number. Here the 6 cells are shown, which start from number 3, 4, 5, 6, 7, 8. The near cell, which is the nearby cell where the anchor cell secretes a ligand and activates quickly, is the 6 number cell. 5 and 6 are near to it. Direct in relation is P6P. These are represented by forward P and the backward P. So we write P3P to P8P. Each cell we will represent P3P, P4P, P5P, P6P, P7P, P8P equal to 6 cells. So anchor cell is sending its concentration to these cells. The more concentration will be seen in the P6P, which will be near to the anchor cell. Now this is the diagram of the C. elegans. This is the ventral part, this is the dorsal part. On the dorsal side, this bar of green color in which gonads is written is in direct contact with anchor cell, which is beneath it. So they activate the gonads. In the gonad, the anchor cell is being activated. An anchor cell helps in anchoring processes of vulva and the C. elegans processes. Vulva patterning and morphogenesis takes place through anchor cell only. It is present beneath the gonad, which I told you 2-3 times between the topic. The gonads, what is the purpose of gonad? The green color gonad, which is seen over here, which is present on the dorsal side, produce gametes which will participate in reproduction. Ventral side, 6 VPs, which are called vulval precursor, present in the mid-ventral position. They initiate cells for vulvar formation, P3P to P8P. Here are the 6 cells shown in this groove-like part, which will be activated by anchor cell when it will be secreting ligand, and concentration of that will come here and will activate this. Further about this C. elegans, we will study in the next video. Furthermore, about development of vulva of C. elegans will continue in next video. This is the part 1, vulvar formation. Friends, nothing is difficult in the life. Look and see the video to understand the concept again and again. You can watch the video. Friends, like it, subscribe it, press the bell icon, and have a happy time. Stay blessed, everyone.